So I am Dr. Vipras Kumar Tiwari, presently working in North Bengal St. Xavier's College in the Department of Microbiology, and my research area is also the antibiotic resistance. So it's a topic. This one is also interesting for me. So being the charge of this session, I would like to just introduce this topic to everyone, and then later we can discuss our research so that each participants can share his research experiences, and that will help the students to design their experiments, and also the researchers to design their experiment from our own experiences. So we all know that antibiotics it's the greatest means medical discoveries on the 20th century, and they transformed the modern medicine by enabling the treatment of life-threatening bacterial infection such as the pneumonia, tuberculosis, and the sepsis. And since the development of the first antibiotic, ampicillin, in 1928, it saved the means antibiotics have saved the countless lives, and it made the procedures like the surgery and the transplant more safer, significantly safer. So, but what the problem that the very drug that once gave us control over the infections are now losing their effectiveness. So the world is facing a crisis, and that crisis is the antibiotic resistance. So a phenomenon in which bacteria evolve and they become resistant to the drug that means to kill them. So once the antibiotic resistance, we all know that when the bacteria develop the mechanism to survive in the exposure of the antibiotics. So now it's important to discuss that what are the reasons for the emergence of the antibiotic resistance and how can we control the emergence of the antibiotic resistance. So we all know that this antibiotic resistance is not the new phenomenon. So it's the natural evolutionary process. But the misuse and the overuse of the antibiotics, we can say they accelerate or amplify the emergence of the antibiotic resistant strains. And the extensive use of those antibiotics in the agriculture or the animal husbandry, not because they're not to use those to treat the sick animals, but also to promote their growth and to prevent disease. So bacteria by different ways can means emergence or the spread of the resistance. We know that by the mutations and also by the genetic exchange, that is the transformation, conjugation, and transduction. And also the bacteria can resist the antibiotics in the various ways. With the mechanism, we know that by producing the enzymes that can deactivate the protein by present of the efflux forms to expel the antibiotics, and by inactivation of the receptor or by modifying the receptor, they can resist the antibiotic that were meant to kill them. So the consequences of the antibiotic resistance are alarming, and we all know that one of the paper published in the Lens in the 2019, there was estimation that about the 7 lakh people are die each year due to the antibiotic resistance strain. And they have estimated that the 10 millions annually by the end of the 2015, 2050. So this antibiotic resistance is one of the most urgent public health threats of our time. And the idea of that entering a post-antibiotic era means before that 1920s. So the minor infection or cuts become deadly is no longer a distant possibility. So I just want to use one quote by the Nobel laureate, means Alexander Fleming, that misuse of the penicillin could lead to the emergence of resistance bacteria. So let us now discuss that how can we isolate the antibiotic resistance strains, or how can you restrict the emergence of the resistant strains, and what are the means methods we can apply to minimize the emergence of the antibiotic resistance. So now this platform is open for the discussion. So one more request that just write your name, designation, and affiliation in the chat box, so the new researchers or the students can contact you for if they have any query in the future. So good
Yes,
evening,
yes,
everyone. So now the platform is
that is
open for
needed.
the discussion.
Correct. So
So one by one,
Vikrant, sir,
the research.
Vikrant, sir,
Yes, sir.
that is very important that all should write down the name.
Sure, sir.
email ID.
Because the
And
objective with this round table discussion, sir, is gathering means bridging the gap that is in between the research scientists and the students.
so there is a Apurva. Apurva, in short, you tell the research in short only.
Yes, sir. Thank you very much for giving me a chance to describe about my thesis. Actually, my topics was isolation and characterization of multi-drug resistant non-fermenters. In my research, focusing organism or prevalence organisms was Pseudomonas, Acinetobacter, Alkaligenous specialis, such type of organisms. And I was trying to find out the virulence genes and prevalence of virulence genes and horizontal gene transfer, whether the resistant gene is encoding by plasmid or chromosome DNA. And then I was finding what is the actually relationship between ESBL, MBL, metallo-beta-lactamase producers, as well as correlation with the efflux pump producers. So I was finding and isolating more than 100 numbers of antibiotic resistance organisms and find out 50 numbers of organisms. They are showing multiple resistancy having different type of virulent genes, and most of them are positive by NDM producer, within the metallo-beta-lactamase, and showing resistancy against almost all of the multiple drugs except colistin. So in such cases, it was very problematic and limiting the therapeutic options, and that was very critical and alarming. So these are also correlated with the production of different additional virulent genes like efflux pump productions. The genes involving for the pump production is ASC6 LBCR as well as Q1 and D. So this such type of genes that presents in organism like Acinetobacter, Alkaligenous, then Acromonas, then Pseudomonas. Of course, Pseudomonas is the major prevalence organisms are associated with different type of infections, but mostly they are involving for the production of PAS or UTI infections. So that was very critical situation in India. Being I was completed my thesis in 2018, and that time the prevalence was 17 to 80%, 18%. That is alarming. And I found that was horizontally gene was being transported from one strain to another. So such type of situation is alarming. But right now I don't have any scope to continue this research project again. But it is right now to discover something, because I know there's a lot of studies are done on antibiotics, but it is time to prevent the resistance and prevent the prevention of resistance and infection controls. So many research are nowadays being going on, like on silver nanoparticles, or we have to design any new drugs molecules against resistance genes. But my findings is NDM, which is the very common and very tremendous virulence genes that produce multiple type of antibiotic resistancy, and it's also helped to coexistence of another genes like efflux pump producer, quinol resistant gene, aminoglycosides resistant genes. That's all. I have published it many paper, and I still now I am working 4 papers related to resistancy. And in Bangladesh right now, my working place is in Bangladesh, and the same prevalence rate is having here. So that maybe the resistancy, maybe it is from different sources. So I found lot of poultry producers are using tremendous level of antibiotics, and now it was very alarming, and I was very astonished. Out of 10 patients, the 10 patients having resistance against ciprofloxacin and levofloxacin. To find it out the cause and reason, I was find it out it in Bangladesh. Most of the poultry and fish producer farmers are using ciprofloxacin. So that is alarming. But my concern is here, and this is very related to my thesis topics. But I am looking to get something developed to control the resistant genes and new drugs molecule. Even right now, in last 1 month ago, we are finding 10 isolates that show complete resistance up to 264 microgram per ml. They are showing resistant against imipenem and meropenem. These are the carbapenem type of drug being used to save the life. This is considered as the last choice of therapeutics. So organisms are right now showing resistance against meropenem and imipenem, which is alarming. So I expect someone or our followers or our students or our colleagues will conduct research to control the resistance genes or any type of analog drug. And also there is very good choice of drug is peptides. Nowadays, as many synthetases are start working. So my suggestion or my request, if anyone, though India is a very big and versatile country, and I respect Indian scientists, I know that you guys have a lot of potentiality. So it's time to think about to conduct research on control of antibiotic resistance rather than finding of virulence genes, prevalence. That's all. That we had done it, and many scientists have done it. But right now we have to find it out something different to save the life. That's all. Thank you.
Thank you, sir, for sharing your experience to us. So now if you have any query to sir, you may ask now. Hello.
Anyone can ask if you
Anyone can
have
ask
any
if any
question
query
or
regarding.
if you need
Yeah,
any
what type of
support.
microorganisms or the bacteria you were looking for this antibiotic AMR, and then what was their source? Can you elaborate on that?
Actually, sir, my findings was on and non-fermenters. Like non-fermenters mean those organisms which are not capable to catabolize carbohydrate, or they are using different energy source other than carbohydrate. And most of the prevalence organism belong to groups are Pseudomonas, Acinetobacter,
Okay.
maybe Acinetobacter baumannii, Acinetobacter calcoaceticus, Alkalizinus picalis, this type of organism. I was preparing this chapter from Kuhneman Color Atlas of textbook of microbiology. There is a chapter number 7. My focusing chapter was there because of their slow growing. See, Acinetobacter baumannii. See what happened in Bangladesh and India. Suppose you are going to submit your sample to any laboratory staff to check the sensitivity. They were sticking on plate and wait 24 hours up to 48 hours hardly and show the resistant pattern. But from my experience, we found it these organisms are very slow growing. Even they are taking sometimes 72 hours to 4 days for the growth, like Acinetobacter, one strain, Acinetobacter calcoaceticus. So these organisms, though it is not identified properly in laboratory, as well as they are slow growing, and the problem is they are showing multiple drug resistance. Even they are showing resistance against meropenem and imipenem. That is very challenging. So my research topics and findings was such organism, those who are non-fermenters. And basically the prevalence organism was Pseudomonas, 80% was Pseudomonas, and 13% was Acinetobacter. But the problem is Acinetobacter is very dangerous organisms and having multiple virulent genes, NDM, CTXM, VIM, this such type of gene, even associated with different gene like C6LBCR, QNAD. So I found in one organism, 7 virulent genes were present, and I isolate 25 kb weight of plasmid DNA, and that organism showing resistance against all of the drugs except cholesterol.
And what is the, according to you, is the new finding? Acinetobacter, Pseudomonas are otherwise also known for a nosocomial infection, right? Acinetobacter baumannii is well known for the carbapenem resistance. Similarly,
Yes.
Pseudomonas also has gram negative.
Yes.
Now,
All
what
of
is
them,
the
non-fermenters, most all of the non-fermenters are gram negative.
Yeah, that is what actually I mean. What was actually the new findings from your study? Or did you see the higher level of the resistance or the genes which are associated with this resistance?
Actually, my target was to find out the virulence genes and prevalence of genes. So my findings, I find out NDM genes, that is the potential genes, New Delhi metal beta lactamase genes. These, if these genes presence in any organisms, it's showing multiple drug resistance, and the relationship between NDM, it is also associated to the coexistence of other genes like AAC-LBCR, which is producing, which is the genes is responsible for efflux pump production, and QNRD type of genes. So the relationship between two virulence genes means NDM never never exists singly in any organism. It must associated with another genes that encoding different type of resistance, and mostly it was related to efflux pump production. And I found the organism showing resistance up to 264 microgram per ml type of concentration of meropenem. That is very alarming. And even it show the, yes, of course, though it is metallo-beta-lactamase producer, so imipenem EDTA was also spreaded by this organisms. And the problem was, I found it out, uh, the virulence genes was horizontally transferred to, I was using that time E. coli JM107 alpha, this organism as a competent cell that was transferred from one strain to another strain and showing resistance against ciprofloxacin, levofloxacin, and also using ceftidizine and cefotoxin.
And what are the resistance which you have recorded are found? Is all are transmissible?
Not
Did
all.
you see they are Did you do such experiment related to the transmissibility conjugation type of experiment?
Yes.
Or the inducible resistance that you use when some
Yes.
other
Yes.
antibiotics of the similar group at a lower concentration, and you
Yes, I did that one.
yeah.
The horizontal gene transfer activity I was finding out, and inducible
Mhm.
resistance was finding out in the in such topics, and I published a paper on that also.
Very good.
Right now I am working, sir. Actually, I am right now working in Dhaka, though it is very related, not related to very enriched molecular biology laboratory, but I'm still now trying to find it out the spread of NDM genes. My research interest is on NDM because NDM is very dangerous, and presence of NDMs really make very challenging to continue the therapeutic options for doctors. So right now I am working on uropathogenic non-fermenters to find it out what non-fermenters are associated for the infection of UTIs, so, and their prevalence of NDM genes. And recent, I am just being prepared one publication and waiting to submit in Willy. So my findings is 13% of non-fermenters are find positive carrying out of NDM genes.
Mm-hmm.
I will be very happy if anyone interested to continue on this particular project of research. But my suggestion, if there is any possibilities, start working development of different peptides drugs that help to prevent resistance against multi-AMR strain.
Exactly.
Because conventional antibiotics nowadays, it is really difficult to find out the therapeutic options. Nowadays what happening, I see the alternative therapeutic options are going on, like you have to take ciprofloxacin with imipenem or meropenem injections. Is 1 gram meropenem injection. Even though this is very challenging, if any strain developed resistance, then it is very hard to treat. And one I find it out, one acinetobacter strain from a patient having cancer in tonsils. So I believe these organisms, so this chronic resistance may train to producer of different type of cancer as well. But because of limitation, because of time frame, I did not able to conduct such type of continuous study because I am from the time I'm from Bangladesh. So I have visa limitations and time barrier. I have to complete my thesis within 3 hours, 4 hours like that. But it will be a very good research project if anyone interested to develop any peptide drugs against AMR microorganisms. So it will be the very current and very hot topics of research.
Now, what you do presently?
Sir, I am right now working as a technical director of a instrument manufacturing organizations, as well as because of to continue my research interest, I am also working as a honorary scientist in a medical college. For actually in medical college, I prepare some research projects, topics to find out fund as well as take care of their molecular biology activities. But my focusing is on in an instrument organizations. So we are selling different type of analytical instruments like PCR, RT-PCR, LC-MS/MS, GC-MS/MS. So still now I am also involving myself with research in medical college as well as taking care of such type of activities in Bangladesh. I am not in education or not in teaching field. Here is problem because in Bangladesh, see that teaching profession is very critical for us to complete study from abroad, because there is one rules in our universities, so they are try to find it out that topper from their own student selections. Their selection procedure is different, so that is why I did not go throughout this educational field. I am right now in industrial activities.
Tiwari sir,
Yes, sir. Thank you so much, sir, for your.
yeah, actually I have to do some work actually, so I have to leave now.
No problem, sir. Thank you very much. And if there is any
Yeah.
option for continue my research, I am ready to left my job and come to join as postdoctoral scientist or any type of work.
Yeah,
Because
I kept my in the chat box. My address is there. Anyone, those who wants interested in the AMR, especially with reference to the food chain, because I am associated with the development of National Action Plan
okay.
on AMR. Yes, and on the antibiotic residues panel of the FSSI. All these discussions, you people expertise, then your research work, is directly useful for our activities at
Thank you.
FSSI as well as at the development of National Action Plan, which WHO India office is actually undertaken that work. So thank
Very
you
good.
everyone. My details are given there already. Thank you.
Okay, sir. Thank you very much. Any other question, please? I am ready to answer my best.
So any question to Apurva sir, kindly. I think, sir, there is no any question to you now.
Okay, thank you.
Thank you, Apurva sir, for your insightful men's experience and your research.
I already shared my details, but I am right now showing my mobile number and email address.
Any faculty member or the scientist who want to share the experience, I welcome anyone. Dr. Avinash Dotto, Dr. Rupa Niroli, Dr. Vishwas, Fatma Barani, Omar, Dr. Vishwas.
Good evening, sir.
Good evening, Dr. Vishwas.
Good evening, sir. I'm Dr. Vishwas Patil. I'm associate professor and head of the department, microbiology department, from Lal Bahadur Shastri College, Satara. I'm a research guide in microbiology from Shivaji University, Kolhapur, and currently 5 students are working for their PhD program. One of the student this year, she has registered for PhD, and her topic is related to the antimicrobial resistance, that is from Western Maharashtra region, for treating the acute infections or gastrointestinal, whatever the most common acute infections the patients are suffering from our region. The antimicrobial, the pathogens from these acute infections, they will be first isolated, and then identification will be done, confirmation will be done, and then the focus of my student is to study the genes associated with the antimicrobial resistance. So Dr. Sarkar sir, he has very nicely explained the all approaches. So I hear with appeal, Dr. Sarkar sir, that in near future, after 1 to 2 years, I request you kindly for collaborative work. So it will give fruitful results.
Thank
The
you, sir.
basic data
Actually,
and everything
Thank you very much.
Huh.
Actually, I am not in educational field or because of few limitation. I'm right now in industry, but could still continue my research work with one medical college. If I will be very happy
Okay.
if I cross and help to your student and you guys or anyone in India if there is any chances for further continuing of research as a foreigner. So I will be there and happy to join them. But for though you are from Kolhapur, I was in Bid district, Bid. I was in Bid and
Okay. Okay. Bid. Yes, sir.
yes,
Yes, sir.
I was in Bid and Aurangabad. I was few of my work was done on Usmanabad as well in the Smuxas lab. So I know the
Okay. Okay.
virulence. You
Okay.
can find it out in my Google Scholar site that the virulence genes in that area, particularly focusing on NDM, CTXM, QNRD, and
Uh-huh.
ASLBCR. And though my interest was very focusing on non-fermenters
Mhm.
like Acinetobacter and Pseudomonas, alkaligenous, so such type of organisms are harbor of such type of resistance genes. So the type of activity I did in my research, and most of the
Okay. Okay.
non-fermenters and antibiotic resistance was associated for the production of pus formation, post-operative patients, hospital infections. I did not find anything from sputums, though it is very common, the non-fermenter associated for the pneumonia production as well as septicemia or such type of abscess. But my core isolation was from pus former, cancer patients having pus, as well as UTI example, urine infections.
Dr. Vishwas, sir?
I was discussing due to poor internet connection, sir. Am I audible, sir?
Yes, sir, you are fine audible.
Hi. Yes, sir. Sarkar, sir.
Yes,
Uh, please put your email ID and contact
I
number in chat box.
yes I already
We
sent
will
it.
go for collaborative work, sir.
Yes I already shared my.
Okay.
My
Okay. Okay, sir.
here. You can find it out in this. Though I was
Okay,
conducting
sir.
research in Bir, Osmanabad, Aurangabad. So the prevalence genes were
Uh-huh.
there is
Uh-huh.
NDM, CTXM, QNRD, and
Uh-huh.
LBCR. These are associated for the
Okay.
production of efflex pump. So I have list of virulence genes
Okay, sir.
and their target and their primer size and their molecular weight. So I have it. If you need, I will share with you. You can order the primers and you can find it out the prevalence.
Yes, sir.
And in
It's
my research work, during my research work, so I find it out the most of the resistance associated with the fast formation patient having fast formation, post-operative infections, and UTIs. I did not find any
okay.
sample
Okay.
having MR associated my non-fermenters from sputums or stool like others. But majority of them are from past post-operative patient having infections and cancer patients, mostly cancer
Okay,
patients.
sir. Okay. Okay, sir. Okay, sir. Thank you, sir.
Yes, thank you.
Dr. Vishwas, sir, you mentioned that your research scholars are working on the genes which are responsible for the antibiotic resistance. So can you, sir,
Yes, sir.
elaborate what type of genes you are targeting, sir? Which genes you are targeting?
Sir, actually, sir, my one of the PhD students, she has registered this year, and just now we have submitted the synopsis. And I was trying to communicate my student to join this discussion, but she didn't receive the call. I tried twice, so she would be talking in more detail.
Okay. See, the resistance actually you cannot target out any genes. So there is a different type of genes involving different class of genes. Like metallo-beta-lactam there is two class A, class B. In class A you can
Uh-huh.
find TIM, BIM, TIM, such type of genes. And in so you have to find it out set of genes. I have all the virulent genes and related primer, related size of primer and how you can
Okay,
identify it. I
okay.
have protocols also. What will be the PCR settings? How you can find it out? What will be your research design? I did that in my
Okay, okay.
area. I have everything in ready state. So if you need any support, any help, I will be happy to help you. And here is my
Okay.
details. I put my two
Okay,
numbers, but
okay. Uh-huh.
it is better to use my personal cell number. I'm just going to send you again my personal details. So in case of anything, you can call me
Okay, okay,
anytime.
sir.
You can send text me. You can email me.
Okay, yes, sir.
I'm happy to help.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir.
And Dr. Deshmukh sir was my means did everything. My thesis corrections, writing. Actually, I was going there to
Sir,
conduct my thesis under his supervision, but that time he was under very close to go his LPR, PRL, so I did not catch him.
okay, okay. Sir, I also proudly say that I am a graduate student of Dr. M. Deshmukh, sir. He was here in Karad at
Karad.
YC College, Karad. I
Okay.
completed BSc under his guidance.
Okay.
Then his guide for everything that I hold the academic position in this academic it's due to Dr. M. Deshmukh, sir.
Very nice.
He's our mentor.
If anyone from Kolkata, I will be very happy to see, because though I am Bangladeshi, but I did my entire of education from India. I complete my graduation from Calcutta University, post graduation from Nagpur,
Okay,
and
okay.
PhD from Aurangabad,
Okay.
Mamu, and then come back again in
Excellent.
Bangladesh.
Excellent.
I know Hindi, I know Marathi,
Excellent, sir. Excellent.
so I will be very happy to see anyone from
Okay,
Kolkata,
okay.
Nagpur.
Yes, sir. Here one
Okay.
Dr. Kirat Kumar Ganguly sir is from Kolkata and he
Ah,
has
sir.
raised his
okay.
hand.
Good.
Yes, sir, Dr. Kirat.
Yeah. Sir, may I continue
Sure, sir, sure, sir.
a little bit? Okay. Sir, nice to meet you. And I heard, actually, I was hearing on the YouTube telecast, and now I joined here. Actually, while you were talking with Dr. Halami over that one, I became interested in that part. Actually, recently in my college, I have isolated a consortium of peptides, which are showing uh, wide range of bacteria. They're inhibiting the growth. They're inhibiting the growth. They're inhibiting the biofilm formation, and this consortium of peptide, I have done LCMS and found the sequences and identified the protein. So it will be very grateful, very helpful for me, if I can contact you and get some future work or some support from your side,
Of
because in our
course,
college
I am also
infrastructure.
expert of LCMSMS, so no problem. I will be very
Okay,
happy to help
okay.
you. Same field of.
We have already done from the ISCB lab, but with these microbial peptides, I want to do some more work, characterization of those things or like that. So in that
Can
case,
you
if I
find it out?
contact.
What is the mechanism? How it inhibit
Yes,
the?
sir. I tried a little bit because they are increasing the porosity of the cells that I have seen. Okay, by dye diffusion methods and cell lysis method. So maybe that has to be done in a deeper way, and the other mechanisms also. So in that
From
context, if
which college you are? I am very recently. Maybe next month I will travel to Kolkata. I got visa
really,
finally.
okay, okay. So in that case, I'm a resident of Kolkata, but my college is in Durgapur, near to Kolkata.
I
Okay,
know
it's
Durgapur.
okay,
Yes.
okay. So my college is there. So if we get some time, we can just have a talk together, or
Yes.
we can arrange also a seminar here. Hmm?
No
So
problem.
I'll be in touch with you over the WhatsApp number. Is it your WhatsApp number, sir? What
Yes,
you
sir.
have
It is
said?
my personal WhatsApp number. And
348. Okay, okay, that I'll contact. Thank you, sir. And this is the platform of MBSI. We are already here, and this is the networking system, I think. We get to know each other. Thank you, Deshmukh sir, for giving us this opportunity.
This was the objective of this roundtable discussion, sir.
Yes, sir.
That
Yes,
we can
Bipran sir.
connect with each other by work.
Yes, sir. Yes, the excellent idea it is. Thank you. Thank you, sir. Vikrant sir,
Yes, sir.
there are many other teachers which are having good knowledge, good research of
Mhm.
antimicrobial resistance. They have joined. So
give opportunity to everyone.
Sure, sir.
Dr. Vivek Upasani is also there.
So Vivek Upasani, sir, if present here, sir. So kindly share your experience, sir. Here. Vivek Upasani, sir.
And many others are there.
Anyone who wants to share
Research.
his research here, kindly raise your hand so that Vivek Upasani, sir. Yes, sir.
Dr. Datta is there.
Yes, good evening.
Good evening, sir.
Deshmukh sir, good evening.
Good evening. Good
This is
evening.
Vivek Upasani.
Yes. So you are providing a service for AMR.
Yes, we are
So
recently
please tell in
we
brief.
do yes, we do whole genome sequencing, as well as if you want to amplify specific genes, we do that. We design primers and various types of, if you want to have the consortium study, metagenomic studies of any sample, clinical sample also we do. Recently, we published a paper in which we isolated a Pandora nemosa strain from a vaginal sample, clinical sample, and we found that it has got antibiotic resistance, and also it is the first opportunistic pathogen that has been isolated from this genital part of, and that is affecting pregnancy also. And so such type of research also we carry out when we get, if there are novel isolates and do whole genome sequencing. And we have also put it in the gene bank, the project details also. And the paper is available online now. Any questions?
Thank you, sir. So if any question to Dr. Vivek Upasani, may ask me.
So, what are the charges for whole genome sequencing?
It depends on the size of the genome, and suppose it's a bacterial E. coli, then it would cost around 15,000 to 20,000, depends. What is the how much GB data that you want, and we that is inclusive of bioinformatic analysis and report. You can say I have given my email ID. You can send your requirement. We give the quotations accordingly, what you are according to your requirements.
Okay, thank you, sir.
Yeah, welcome. We also have students working on such clinical samples. Ours is a molecular diagnostic lab. I would like to tell you that. And because of that, we get different types of clinical samples. And then when you are talking of AMR, then getting clinical samples sometimes is difficult, but we have network throughout the country from where we get the clinical samples. And so if someone is interested in isolating some particular type of organisms, bacteria, pathogen from those clinical samples and finding out the AMR profile and prepare a database, that also can be worked out as a project or for a PhD work. So if anyone is interested, you can talk on this. Yes, over to the coordinator who is there.
Very nice, sir. Vivek, sir.
Yes. So it is an ongoing
Anyone want to give his
collaborate.
research or some comment? Sandeep Yadav is there.
Yes.
Dr. Prasun Kumar is there. Anyone want to make a comment from Nepal?
Also, recently we found a beta hemolytic pseudomonas. And we are studying its antibiotic profile also from a clinical sample, again from a vaginal swab.
Okay, okay. So Bipran sir, please proceed.
So here Dr. Avinash Datto sir, Dr. Rupa Niroli, Dr. Vishwas.
Good evening, sir.
Good evening, Dr. Rupa.
Yeah, good evening, Deshmukh sir. How are you, sir?
Good evening, good evening. I'm fine. So please introduce yourself.
Yes, sir. Yes, sir. Myself, Dr. Rupa Niroli, recently I completed my PhD and I'm a proud student of Dr. A.M. Deshmukh, sir. I completed my MSc from YC only and PhD from Gulbarga University, Karnataka. And my topic of research was antimicrobial resistance. Briefly, let me explain you my topic. So I isolated E. coli from fish samples from Mumbai and suburbs. So basically, I worked with 16 antibiotics from 9 diverse classes. And I isolated around 60 E. coli isolates. Out of it, many of the isolates were resistant to more than 14 antibiotics, and two of the isolates were only sensitive to imipenem. And I have used cefotaxime, ceftazidime, like imipenem, which can be called as last resort antibiotics. So the results were really scary, and I focused on beta lactamases. And my main focus was on beta lactam antibiotics, and I have isolated three genes from the isolates, that is CTX and SHV and TEM. These were the three genes I isolated, and I conducted PCR studies for the same. And one more thing I just wanted to mention, that this is actually a proud thing, that we worked on the solution as well, our lab definitely. One of my colleague, lab mate, that is Dr. Narmada, has worked on it. Like she has isolated some bacteriocins from Lactobacillus plantarum. These two strains which I'm speaking about were sensitive to bacteriocins, which we have worked on, and it has shown a very great zone of inhibition around 17 millimeter, wherein it has shown imipenem sensitivity also around 7 to 10 millimeter. We have found a solution for that, and we worked on bacteriocins as well. So this was a brief introduction about my topic, and if any questions are there, I welcome them. Thank you, sir.
Thank you, Dr. Rupa madam. If any question, now the dash is open for the question. So there is no any question.
Thank you, sir. Sir,
Thank
one
you.
more thing, I just want to congratulate sir, that you are, I mean, this is a great initiative wherein the students who really wants and interested to work in antimicrobial resistance, they'll be finding good guides here. So, which is very rare event, like, because during my PhD, I really struggled to get the information from the researchers who has worked and who really want to help us. So really, this is a great initiative and great platform. Credits to Deshmukh sir. Thank you, sir.
Thank you so much.
I have a question. You mentioned about the sensitivity. You said 17 mm, right?
Yes, yes, it is 17 mm. Yes.
Okay. Okay. So, and you have identified the bacteriocin also?
Yes, yes, yes. We have conducted studies on the bacteriocins. Uh, Many studies have been conducted, and we have published papers also on that.
Okay. I would like to go through those things. Maybe if you
Surely,
can share some the soft copies of those
surely
papers.
you
I'm
can.
Haresh Keharia. Yeah.
Okay, you can
Okay.
connect with
And
me.
yeah. Okay.
Yeah,
Sure. Thank you.
yeah, yeah.
So any faculty members who want to share now experience on antibiotic resistance? Joel Kanbi, Farah Barmi, Asmita Bambole,
Actually, I want to introduce my research work. I am not exactly working on antibiotic or antimicrobial resistance per se, but I am working on development of direct fed microbials to enhance the growth in poultry, and the objective is to use as a substitute to antibiotic growth promoters. So that is the area I am working on. Already have one PhD student has completed the work. We have contributed to good number of isolates, and it was industry sponsored project. So we have isolated organisms which can work as substitute to antibiotic growth promoters for broiler uh, uh, growth, and it has been validated on commercial animal trials also. However, I'm interested in understanding the mechanism, and that is my current interest. I'm also continuing my interest in isolating more such organisms which can be used as direct fed microbials. So this is the area I am working, and would be very much interested if anybody who is working more closely with animal trials would like to collaborate, or even with cell cultures. I would like to collaborate with them.
Thank you, Aristelia. So from Nepal, I think that Pramila Parajuli Madam has joined.
Yes, good evening, everyone.
Good evening, madam.
It was so interesting because I am planning to join my PhD and I might be joining in AMR related topic. So I joined here to know about what will be the projects or things. So I just having some ideas what can be done in future. And Harish sir he explained about like growth promoting organisms in poultry. It sounds so interesting. So can you say some organisms or name some organisms that can act as a growth promoter in poultry, sir?
Yeah, actually, there are two classes which people focus on. One is lactic acid bacteria, and the other is bacilli. We had focused on bacilli as substitute to antibiotic growth promoters. It was because we were having this as an industry sponsored project. Jitex Biotech had sponsored the project, and we had isolated bacillus strains. One of the isolate was Bacillus valegensis, and it had antimicrobial activity against most of the gut pathogens that we tested. And then this isolate was characterized for its probiotic traits. It passed in that, and this culture was then used uh, as augmentation to animal feed, and it showed very good results. It showed better results than the antibiotic growth promoters, and it also exhibited immunomodulation. Study the mechanism in detail. So that is currently the interest.
Yeah, it sounds so good. I also work in antibiotic resistant organism from poultry. I received one grant from University Grant Commission, and we have tested 1,000 sample and for colistin resistant isolates. We have the huge number of colistin resistant E. coli. We were focusing on colistin resistant E. coli. So almost 71% of the isolates were colistin resistant. So antibiotic resistant is a huge problem in Nepal as well.
Okay, so in that regards, I think we can test our isolate for its ability to inhibit E. coli, because our isolate completely removes E. coli within 24 hours in liquid co-culture when we inoculate both of them at equal density. And then this can be done, taken up for even animal studies.
Oh yeah. Thank you so much, sir.
Yeah.
Thank you, Pramila. Anyone who wants to share or put any comments on the antibiotic this topic? Dr. Sandeep Jadhav sir is here.
Hello, good evening to all.
Good evening. Good evening, sir. You are audible, sir.
I just only joined to hear the topic. Actually, my research work is related to the bioinformatics and virology. In the last
That's
week, I discussed. So I just want to uh, hear from, just want to listen the topic of antimicrobial resistance because my research work is not related to that. So just for the information, I joined.
Thank you, Sandeep, sir.
Thank you, sir.
Sujata Kuruchka, Tanvir Kaur, Kumari Pallavi, Jatin Gupta, sir. Paresh Kheria. So, if not the research experience, you have any comment, you want to put any comment on the antibiotic resistance, you are mostly welcome. Even the students can share his comment on this topic. Any student, if there is present, you can put any comment on this topic. JY Maniyar, Asmita Bambole, Jatin Gupta, Kanve, Kartik, Kumar Pallavi. Farah Barami, Omar M. Kude.
Hi, sir. I'm Farah.
Yes.
I just wanted to share what it's a small project which we have done in our labs. Actually,
Yeah.
as you said before about the due to the overdosage and overuse of antibiotics, the antibiotic resistance are being taken place. So we did a small project like using combination therapy. So for that, we actually did. We were checking for the synergistic activity, which is basically we are, combining two drugs of different mechanism of action. Because if you target different sites, you can reduce the antibiotic resistance. So we did a lot of combinations, where, taking on the basis of different mechanism of action, and we checked for the minimum inhibitory concentration, and we tried using combinations using this minimum inhibitory concentration as well as using the half amount of the minimum inhibitory concentration. So that is, I think that is one mechanism where we can reduce the amount of drug concentration, and we can, create or create new formulations so that these antibiotic drug resistant organisms don't come into play again. So that was a small work. So I just wanted to share.
Thank you, Dr. Farah. So just have come to the closing time. So anyone who wants to put any comment on this topic? So I'm going to share one feedback form, not feedback form, and just to collect the mail ID so that we can issue the certificates who have participated here. And also the feedback so that we can improve in the chat box. I have shared one Google feedback form. Also put a suggestion that how can we make it more interactive, this roundtable discussion. So just take your 1 or 2 minutes it will take fill out this form. Form is accessible to all of you. Hello. So with this, we come to the conclusion of the today's session. So we can conclude, sir, now. Hello. Deshmukh, sir.
Hello.
So we may conclude now, sir.
Yes, you can conclude. You can conclude.
Thank you, sir. So I sincerely thank each one of you for your presence and attention and engagement throughout the lecture. And I hope this session has grow your curiosity and deepen your understanding of the topic, the antibiotic resistance. My heartfelt thank to all the participants, faculty members, students, organizers, all India President Dr. M. Deshmukh sir, who made this session possible and meaningful. So your time and participation are truly appreciated. So thank you everyone for participating in the round table discussion. So just take your 1 or 2 minutes and fill out this form. Yes, Tejaswini Shinde madam, we will conduct the session on every Tuesday on the different topics. On the last Tuesday, we have conducted on the virology and this week antibiotic resistance. On the next Tuesday, we will conduct session on the different topic. On each Tuesday, we will conduct the session. We will share our link in the WhatsApp group of MBSI. Are you member of any WhatsApp group of MBSI? Tejaswini Shinde. So I will share the WhatsApp group link with you. You can join. From which state you are? Hello. Okay, Maharashtra. No? So I just share my WhatsApp number. Okay, I will share. You can share the WhatsApp number so I can add you in the Maharashtra group by taking permission from the M. Deshmukh sir. So you can get the link every Tuesday.
Sir, I just want to know, do we have a recording of this roundtable discussions?
Yeah, we record.
So I would like to get access to the recording of probiotic roundtable discussion. You had a discussion. I had missed that actually.
Okay, sure. I will.
So please do share with me. I am
Sure.
Harish Kumar Keharia, and
So just send me one hi message. I share my WhatsApp number there. 8670725693.
okay, sir. Sure.